So thank you very much for the invitation. I mean, coming to Cambridge as the cradle of biology at the molecular level is always wonderful. And then speaking in the Max Perlitz lecture theater is, of course, magic in itself. And standing on these shoulders of giants, I would like to tell you before lunch briefly a little bit of the work that we've been doing using single molecule spectroscopy on the interactions of intrinsically disordered proteins. And so you've already heard a lot of work on structured proteins, but also a lot of dynamics today. Let me just mention briefly, for those of you who are not so familiar with intrinsically disordered proteins, that we know now that if we look at our proteome, that only about a third of our proteins are fully folded and structured. The large majority of proteins in our proteome contain both folded parts and disordered regions. And then there's also a small but significant fraction of proteins that are completely disordered. And so it's these kinds of systems where crystallography and so far also cryo-EM and AlphaFold have only been of limited use. A key method, of course, that has put many of these proteins on the map is NMR. But I would like to tell you about another method that we have been using over the years to study these systems, and that's single molecule spectroscopy. But I would also like to mention that it's very clear now that these disordered regions are not just some leftover evolutionary crap, but they're actually functionally important. And so many of these intrinsically disordered proteins are involved in cellular interactions. And one of the most famous examples is shown here, the tumor suppressor P53, which is known to bind to more than 100 cellular targets by means of its disordered regions. And so disordered proteins are very important for function. So studying them in more detail is certainly a worthwhile effort. The method we use is single molecule spectroscopy, and I don't have to tell you how FRET works. You've heard about FRET, especially from Beat now. One point I would like to mention, because this is also a little bit about technology or techniques, Beat was focusing on measurements using TURF, where you have the molecules immobilized on a surface. And the big advantage of this approach, of course, is, as he showed us beautifully, that you can look at many molecules at the same time. Right? So you get an enormous amount of data from your system, and you can look at them at the same time. We use confocal single molecule spectroscopy, and so the idea here is that you focus your laser beam to a focal spot, and you detect from this confocal volume your molecules. And so we can either focus into the solution and then just have the molecules freely diffusing in solution, and if they are sufficiently low concentration, every molecule that passes by generates a fluorescent burst that we can distinguish. We can also mobilize the molecules on the surface and then focus on them individually, but in contrast to the TURF experiments, we have to do this sequentially. We have to scan the surface and then look at one molecule after the other, which can be a little more tedious and limits some experiments. But the big advantage of this approach is, in particular, that we use time-correlated single photon counting, right? So in contrast to camera-based approaches that usually give you a time resolution down to a few milliseconds, in this case, we get the arrival times of photons with picosecond time resolution. And then because of this, we can measure fluorescence lifetimes, we can get time resolved anisotropies, and we can do correlation spectroscopy down to very short times. And I would like to illustrate a little bit how we can use this methodology to study the heterogeneity and dynamics of intrinsically disordered proteins and their interactions. But again, to sort of give you a little bit of an overview of the kinds of timescales that we can address, I would like to remind you that, of course, one of the main challenges in studying biomolecular systems is that we have to cover a huge range of timescales, right? So with single molecule spectroscopy, we can go down to the nanosecond timescale, and I'll show you the dynamics of IDPs are on this timescale, and vibrational spectroscopists will tell you that even faster timescales are important for understanding biology. We have structure formation in proteins, binding, many cellular events that happen on timescales, a very broad range, and some of the slowest processes like aggregation can happen on timescales of a human lifespan, right? So we really need to understand many, many timescales. And single molecule spectroscopy, or the toolbox of single molecule spectroscopy, is now able to cover a very broad range of these timescales. And I will not go into the details. Let me just mention some of the method development that we've also been doing over the years. So for instance, we've been employing rapid microfluidic mixing devices that allow us to probe down to millisecond kinetics using single molecule spectroscopy. We can also measure such as long trajectories, similar to what Beat showed you, but one by one, unfortunately. But since we use single photon counting, we can also push this to very short timescales and go to microseconds. And one method, one aspect I want to point out is also that we can do some of these experiments also inside live cells, by micro-injecting labeled proteins into cells, and then we can position our confocal volume in a cell and get information about conformations and dynamics of molecules in cells. And one aspect I want to highlight in particular, because we will need it later on during the talk, is this method here that we refer to as nanosecond fluorescence correlation spectroscopy. The basic idea here is not only to get information about the average distance between two fluorophores that we attach to our molecules of interest, but to also measure very rapid distance fluctuations, right? So these fluctuations we can pick up with correlation analysis. And so this is an example of such correlation functions here in red and green, the acceptor and donor autocorrelation functions, and in blue, the cross-correlation function. And it's the decay time of these correlations that tell us about the time scale on which the distances between the fluorophores change. And so it turns out that for intrinsically disordered proteins, we are talking about time scales in the range of tens of nanoseconds or hundreds of nanoseconds for large-scale changes in the configuration of these polymer chains, if you want to think about them in terms of polymer dynamics. And just for the people among you that are interested in method development, let me also mention that recently we've started to use nanophotonics, which allow us to enhance the fluorescence. Here we're using these zero-mode waveguides. Also in collaboration with Philip Tinnefeld, we've been using a different technique. The basic idea is you enhance the fluorescence, and with this we can measure even faster dynamics than without. And so there is also still a lot of technology development in single molecule spectroscopy to address these kinds of questions. But now what I would like to do is to illustrate some of these fast dynamics, or a potential to investigate these fast dynamics with a particular system that we've been investigating in great detail over the last few years. And it's these two intrinsically disordered proteins. You've already heard about one of them, histone H1. So this is the histone that sits on top of the nucleosome, the linker histone, and it contains a little folded domain, but large parts are disordered. If you look at the sequence, you can see it's full of lysines, and as an end result, it has a net charge of plus 53, highly positively charged, intrinsically disordered protein. And the other protein is its chaperone by the name of prothymosine alpha, and this is another remarkable intrinsically disordered protein. If you look at the sequence, you see it's full of acidic residues, and as a result, it has a net charge of minus 44. Right? In contrast to histone H1, which has a little folded domain, it's completely disordered and doesn't really form any structure in any conditions we know of. And it's been known for a long time that these two proteins actually interact in the nucleus. And I'll show you a little later what prothymosine does with histone H1. But the starting point for us was the question, what happens when these two proteins interact with each other? What does the complex of these two proteins look like? Then in a collaboration with Dirk Graveland in Copenhagen, and for the NMR and simulations with Robert Best at the NIH, Cambridge alumni, by the way, together with single molecule FRET, nanosecond FCS, and circular dichroism, we found that these two proteins remain disordered in the complex. There's no formation of secondary or tertiary structure when they bind each other. And this is a coarse-grained simulation that is in agreement with all of the experimental data that sort of illustrates what's going on. And what's remarkable is that these proteins bind to each other very tightly. The affinities in the physiological ionic strength range are in the picomolar to nanomolar range. So it's very close, a very strong binding, but they remain disordered. And so in blue you have H1, in red you have prothymosin, and the blob here is the folded domain of H1. It always looks like it's important because it's always in the center tethered between the disordered regions. We can actually chop out the folded domain, and the affinity is still in the picomolar range. So the question was, how does this type of interaction affect the functional properties of such proteins? And so the first question was, how does it affect kinetics? And so I will only summarize very briefly some of the key findings before I go into more recent work that I would like to show you in more detail. And I'll illustrate this just with a molecular dynamics simulation. And these simulations are all based on experimental data, but they nicely summarize the bottom line of our findings. And so the first question was, what happens now if you have such a complex? And now what Andrea Sottini, a fabulous PhD student in the lab, found is that you can not only have one-to-one complexes, but you can imagine if you have these highly disordered systems, it's relatively easy for another protein to come in and join the dance. And so Robert Best simulated this based on our experimental result. And so this is the starting point, this histone H1 bound to a copy of prothymosin alpha, and then he put another one into the simulation box, and you can see now they share the H1. They form what we call a ternary complex until one of the prothymosin alphas dissociates. And so this is a process that was actually observed in simulations already quite a while ago on synthetic polymers by Peng and Mutsukumar, and they termed this competitive substitution, which I think describes this process very nicely. So essentially, one prothymosin kicks another one out. And so the result of that is that not only, you can imagine these two proteins also bind to each other very tightly, so the on rates are diffusion limited, but you can also have this very rapid exchange. So in a way, you can have very high affinity on the one hand, but still rapid exchange between binding partners, which is usually something that you wouldn't see with most folded proteins. Now, this is just prothymosin alpha and H1, but the question is, does that have any implications for what these proteins do in their cellular environment? And so these are results from Peter Heideson from the experimental side and Davide Mercadante from the simulation side. Peter is now professor in Copenhagen. Davide runs his own group in New Zealand. And so now the question here was, what happens in the nucleus, right? And so here you see, again, a coarse-grained simulation based on the experimental data. Here we have histone H1 sitting on top of the nucleosome here on the diad axis. And actually these nucleosomes, Beat helped us greatly to get these up and running in our lab. And so now the question was, what happens when prothymosin alpha comes in? And indeed it turns out, since the large part of H1 remains disordered on the nucleosome, prothymosin alpha can bind to H1 while it still sits on the nucleosome. So again, we have such a ternary complex that's being formed. And so prothymosin essentially neutralizes the interactions of the histone tail with the DNA, and in that way accelerates dissociation of H1. And experimentally, we find an increase in the dissociation rate by about 2 orders of magnitude by prothymosin alpha. And so in this way, prothymosin alpha accelerates dissociation to timescales that are biologically useful, because it turns out H1 alone binds to the nucleosome with femtomolar affinity. So spontaneous dissociation would be much too slow, and prothymosin alpha is not the only histone chaperone, right? So there is many histone chaperones, and this is an example for the function of such a histone chaperone. So these are 2 examples how this type of interaction between disordered proteins can really introduce sort of new interaction mechanisms that we may not have thought about as much before. And so the question that I would like to focus on for the rest of my time is another aspect that is very common for many of these intrinsically disordered proteins, which is that they phase separate at sufficiently high concentrations. And I'm sure many of you have heard about phase separation in the cell as a mechanism that has now been recognized as an important regulatory aspect. And there's some leading work in this area done here in Cambridge. And so, especially in the nucleus, you have many of these membrane-less organelles that are important for processes like ribosome assembly and the like. For many of them, we don't know the detailed function yet, but it's pretty clear that many of them are of functional importance. And so the question was, what happens now with our two proteins, H1 and prothymosin alpha? And if you talk to a polymer physicist or a polymer chemist, they will tell you, I've heard about this, right? If you take two oppositely charged polymers and you put them together, they can phase separate in a process termed complex coacervation. And so the question was, is that something that H1 and prothymosin also do? And might this allow us to get insight into the molecular behavior of these systems inside such condensates? And so indeed, if you use them at sufficiently high concentrations, then they form these droplets of very high protein concentration. Inside these droplets, we have protein concentrations of about 200 milligrams per milliliter. And outside of the droplets, you're talking about concentrations in the nanomolar or micromolar range, depending on salt concentration. And if you measure the viscosity of these droplets using microbiology, by bead tracking essentially, then what you find is that the viscosity in this case is about 300 millipascal seconds, so 300 times more viscous than water. So very viscous entities, and I would like you to remember this number of 300 millipascal seconds because we will need it later on. The key question that we wanted to ask, and this is a project that Nicola Galbanetto, a brave and fabulous postdoc in the lab, took on, was to see whether we can measure the conformations and dynamics of molecules inside these droplets, right? So the idea was phase separate the two proteins by just mixing at a sufficiently high concentration. You get these droplets, and then you dope them with a little bit of labeled protein, right? So that we can do the Fred experiment on the individual proteins inside the droplets. And so if you now place your confocal volume, we can decide whether you want to measure the dilute phase. And if you measure the dilute phase, this looks like these free diffusion Fred experiments typically look like, right? So every burst of photons that you see here comes from an individual molecule diffusing through the laser beam. And you can analyze these bursts as individual single molecule events. And of course, you can also place your focus into one of these droplets so you know exactly where you're measuring. And so then what you see here from Nicola's data is you can also get these bursts of photons, but if you look at the time scale, they're much longer because now we have this highly viscous environment, and so diffusion through the focus takes much longer, right? So that's already a first signature that you see here. It turns out these experiments, we've been trying to do such experiments for almost 10 years. They're very challenging. The main challenge is a very trivial one, actually, which is that if you imagine you have protein concentrations of hundreds of milligrams per milliliter, the slightest impurity in your protein preparations will make such a single molecule experiment impossible. So in this case, it turned out that we had sufficiently pure preparations so this experiment could be done, and Nicola got it to work. And so now we can compare three different situations, right? We can compare prothymosin alpha, which we labeled here, just by itself in dilute solution, or prothymosin alpha bound to histone H1 in this 1-to-1 complex, and prothymosin alpha in a dense phase with its partner H1 inside the droplets. And so first result would be transfer efficiencies. Prothymosin alpha alone, in this case, we have transfer efficiency of 0.35, which considering the sequence separation of the two fluorophores, tells us it's a pretty expanded chain, right? Because highly negatively charged, a lot of charge repulsion, so the average distance between the fluorophores is relatively large. If you now bind it to the positively charged histone H1, then the transfer efficiency shifts to a higher value because prothymosin alpha becomes more compact when it's hugging H1, right? So this is what we already knew from before. Now the question was, what happens in the dense phase? And as you see here, the transfer efficiency is right in the middle, right? So it's more compact than by itself, but it's more expanded than in the one-to-one complex. And as you might guess, the reason is that inside the droplet is now not just interacting with one H1, but it's interacting with multiple H1 molecules at the same time. So it needs to stretch out a little more. But the real, so from this we know something about the average conformation of these molecules inside the dense phase, and it turns out by using information from the fluorescence lifetimes, we can also tell that under all these conditions, the protein remains disordered. Under all these conditions, it's sampling a broad distribution of intramolecular distances. The real question was, what about the dynamics under these different conditions? And so again, for the first two conditions here, we already knew the result from previous work. So if you look at prothymosin alpha alone, and so here you see this very rapid decay in the nanosecond FCS experiments that I mentioned in the beginning. The time scale is about 14 nanoseconds, right? So that's the time scale on which this little protein polymer reconfigures or changes its configuration very, very rapidly. If you bind prothymosin alpha to H1, you already see a slowdown of these dynamics by about an order of magnitude. We're now in the 100 nanosecond range. Still pretty quick, but significantly slower. And so the key question was now for us, what happens in the condensate? Right? Now remember, we had a viscosity of 300 times greater than water. So the question was, would the dynamics also be 300 times slower than in this situation? And the surprise was that it wasn't. And so if you now look at the change in time scale, you only get about a factor of 3 slowdown inside the condensate. And so this was the key question that we had to answer. Why can the dynamics of the chain be so fast, even though the bulk viscosity is so high? And so there are two components to the answer. I only have time to tell you about one of them, but if you're interested in the other, I'll be happy to tell you later on, or in the discussion. And to address this question, Milos Ivanovic, a fabulous postdoc with a simulation background, he took the challenge of running a large-scale molecular dynamics simulation of the system. And now, since we wanted to compare absolute timescales, he had to run an all-atom explicit solvent simulation. So what he did is, he put almost 200 of these proteins in a box, including all of the solvent and all the ions. This ends up with about 4 million atoms altogether. And then he ran it on the Swiss supercomputer for 6 months, and he got 6 microseconds of simulation time. So this might sound pathetic to you, but since we knew from the experiment that the dynamics we're looking for on the time scale of a few hundred nanoseconds, we thought it's worth a shot, right? I mean, we might get enough simulations to actually get reasonable convergence on the time scale that we're interested in. And so this is a movie from such a simulation where you can see in red prothymosin alpha dance within the dense phase. So this is just one of them highlighted with a few histones in different shades of blue. Now we can compare simulation and experiment side by side. We can compare the protein concentrations. We can compare translational diffusion coefficients. We can compare transfer efficiencies, and they all agree remarkably well, I must say, better than we would have dared to hope. And most remarkably, the reconfiguration time, so the chain dynamics that we see in experiment and in simulation, are fabulously close. Right? So this gives us some confidence that it's maybe useful to look at these simulations to understand what's going on at the molecular level. Why do these dynamics remain so rapid? And so let me just show you 2 key results that Milos got from his simulations. The first one is here. just looking at the number of interactions that every residue in the prothymosine molecule makes, right? So this is now just going along the sequence of prothymosine alpha and just counting the average number of contacts that every residue makes. And in gray, you have the one-to-one dimer, and in purple, you have the dense phase. And you can see it's pretty similar. Right? So that means that the individual molecule of prothymosine alpha sees a pretty similar environment, whether it's bound to a single H1 or whether it's in the dense phase with all of these other molecules around. Right? So the local interactions are very similar. And if we now look at the time scales, even more interestingly, right? So what Miloš did here is he quantified the time scale of interactions between residues, right? So especially of interest here is the lifetime of the salt bridges between the charged side chains, of course, right? The lysines and the glutamates and the spartates, and seeing how long lasting are these salt bridges, for instance. And the remarkable result is that these contact lifetimes are in the low nanosecond range. So there are no salt bridges that last for microseconds or milliseconds, but these salt bridges are very, very short-lived, both in the one-to-one complex and in the dense phase. And the question is, why is that the case? And so if we now look at the simulations at this very short time scale of a few nanoseconds, we can try to understand what's going on. And first I would like to remind you of our very high protein concentration. And if we just count the number of charged side chains, then you get an average concentration of charged side chains inside the droplets of about 1 molar. Right? So there's always a charged side chain in the neighborhood of any of these interactions. And then the kinds of processes that can occur is something like this. So imagine here we have a situation of lysine 160 interacting with glutamate 80 and forming a salt bridge. But then here comes another lysine residue, interacts with this salt bridge. So again, we have this concept of a ternary complex that can form in these highly charged systems. And what it does, it sort of kicks out the original guy. And so one salt bridge can replace the other. And you can imagine now with this enormous density of charged side chains, this is something that can happen throughout the system very rapidly. And I think that this kind of behavior is likely to be relevant for interactions of such highly charged systems of polyelectrolytes, especially in the nucleus, where we have this enormous density of charged molecules. And I think this may be an important contribution, how the nuclear interactions remain dynamic, even though they need to be very strong in many cases. And so we are now going into directions where we're trying to vary the sequence. We're looking at other molecules. So for instance, you also find molecules like protamine that is important in packaging DNA in sperm. We have lots of arginines. It turns out that this slows down the dynamics a lot. I don't have time to talk about this here now, so this is for another time. But those of you interested in phase separation, you might want to have a look at Nicola's latest preprint, where he investigates the influence of these different sequences together with Miloš in the simulations. And so with that, I would like to summarize with a little phase diagram, where in this phase diagram, what's shown here is on this axis, the concentration of prothymosine alpha, a negatively charged IDP. On this axis, the concentration of histone H1, a positively charged IDP. And so in much of this phase space here, we form the dimer of the two molecules, right? So that's what we started out with, this highly dynamic, high affinity complex where the molecules remain retained at this order. In single molecule experiments, we can even go to low picomolar concentrations so that we can also study the process of association and dissociation of such a high affinity system. If we increase the concentration of one of the two proteins, and we start to form these ternary complexes, and I told you these kinds of ternary complexes are key for the rapid kinetics and rapid exchange between binding partners, and that this may be functionally relevant, for instance, in the nucleus, where we have such chaperoning of histones by negatively charged IDPs. And then finally, here, if we go to very high concentrations of both proteins, we can form these phase-separated systems, these coacervates, where we have very rapid dynamics inside these droplets, even though the bulk viscosity is very high. And the key contribution here is that we have this very rapid exchange of contacts that are formed. And so with this, I would like to thank the fabulous team. I think I mentioned everyone along the way. Maybe let me highlight again Alessandro and Madeline, both also Cambridge alumni, who started this whole work on the system of H1 and prothymosin alpha. I mentioned Andrea and his work on the kinetics, Peter and Davide on the nucleosome, Nicola phase separation with help from Aritra, who also studied the thermodynamics of this process in great detail, simulations by Milos, and Daniel, a key person in the lab for everything from coding to instrument development, data analysis, and modeling. I would also like to thank Rob, and all of the simulations I showed you were either done by Rob or with a lot of help from Rob in a close collaboration. I would like to thank Beat and Sinan for their help with reconstituting nucleosomes, so we could learn how to do that and get these experiments up and running. And all of our NMR work is done with Beat in Copenhagen and her team. And with that, thank you very much for listening.
All right, we have time for a few questions.
Just wanted to ask is, so you showed that the number of interactions between the dimeric and the condensed phase are similar, but then why do you see a shift in the FRET efficiency between the two?
Yeah, good point. Let me just go back to the slide maybe. So the number of interactions are very similar, but the key difference is that the interactions are made in one case just with one protein and the other case with many proteins, right? So we count here every interaction that a prothymosin alpha residue makes, right? And in the case of the one-to-one contact, it can only make interactions with the single H1 that it's bound to, but in the dense phase, it makes interactions with many different H1s, on average sort of 6, 7, 8 different H1s at the same time. And so you have this network of interactions, and so that's why it's more expanded in the dense phase.
Okay, so in the purple curve, basically it's also the interaction between two things in the condensed phase and not the entire thing mapped.
It's any interaction that any prothymosin residue makes with any other protein.
I see, okay.
Yeah? So not just one neighbor, but all other proteins present.
Okay,
Yeah.
thank you.
Thank you.
Tejun.
Very interesting. But many intrinsically disordered parts of a protein actually, we say it's intrinsically disordered because we isolate protein look at them individually, but they are hunger for partners, want to hug them. And we do see a lot of cases like an intrinsic disorder partner, large part, is disordered, but when we have a partner bound to it, it's extremely well ordered. We can resolve the structure to 2 angstrom resolution. This is one of the cases for the carbazosome shell protein with a linker we determine. Now, do you see similar situation in this case, it's just come and go, never stay there together, seems. But do you see the case there actually stably bound and well structured upon partner binding?
Well, they are stably bound, but they're not well structured. I think that's sort of the surprising thing here. Right? You can be stably bound, but you don't need to be folded. Right? And in this case, of these two proteins, it doesn't look like there is any secondary structure formed upon binding to any partner that we know of. But of course, as you mentioned, there are many IDPs that do form some secondary structure, for instance, or even tertiary structure when they bind to another binding partner.
No,
That's actually something that AlphaFold is pretty good at predicting, right? So if you take an IDP, AlphaFold predicts a helix, it doesn't mean that this helix is formed by the IDP itself, but sometimes this helix is formed only in the context of binding another protein.
No, we found this is not the case, actually. The protein stayed completely unstructured for the second structure, just all random stretches still stay. And we did AlphaFold, they don't form second structure at all, but stay like well ordered in a way when they bond to the partner. Not necessarily have forming secondary structure, but well ordered.
Well, I mean, they need to form some sort of secondary or tertiary structure to be well ordered, I guess.
Thank you. Beautiful work, beautifully explained. The other way to look at this is from the water's point of view. The net solvation of these molecules is not changing. Is that how I interpret that?
Yeah, this is a question I would love to be able to answer. This is something that is really challenging to address, right? So if you look at something like this, right? I mean, even though we have a protein concentration of 200 milligrams per milliliter, that still means that 80% is water, right? So they remain highly hydrated. Still, it is likely that enthalpy and entropy of water will be strongly affected inside and outside. There is another interesting aspect, which is that the binding between these two proteins is actually endothermic. And one of the key driving forces is release of counterions. So you have interesting entropy, enthalpy effects going on, and I'm sure that solvation has an important part to play, but it's been very difficult to address this quantitatively. So if you have any ideas, I'd be very keen.
Well, I was thinking about you were swapping ion pairing partners. It's as though water doesn't ever get involved in that. There's a net solvation, and maybe water's moving from one to the other. No, it's fascinating.
Maybe I'll ask one last question. So because these interactions are non-specific in a way, there are many basic proteins in the nucleus that will interact with one acidic partner. And so how does it actually become specific?
Yeah. So I think this is one of the big questions, how all of these polyelectrolyte interactions in the nucleus can still be sufficiently specific and regulated so we don't just have a big charged blob in a mess, right? And so there are some situations where it's simple to answer. So in many cases, these disordered regions are part of a larger protein with a folded domain. Like, so for instance, for histone H1, the folded domain is important for positioning it on the dyad on the nucleosome. But the disordered region is very important for increasing the affinity non-specifically to DNA in general. And we find this for many DNA-binding proteins and RNA-binding proteins, that they have long disordered tails that modulate the interaction, right? And so another example would be a DNA-binding protein with a positively charged tail that mediates interaction to the DNA, so it can sort of slide on the DNA and remain bound, but not specifically, but it has a folded domain that then, for instance, recognizes a DNA damage, right? And that binds it to a specific spot. And so the cooperation between folded and disordered proteins is certainly part of the answer, but there's of course much more to it, including post-translational modifications and whatnot.
All right, in the interest of time, please join me in thanking all three speakers.